try to explain this highly variable phenotype in Proteo-bacteria. So how is it that these organisms are able to so easily switch from one phenotype to another? Now, we're not talking about individual cells, although they can do that too. But in an evolutionary sense, how is it that you have close relatives where one's a phototroph, one oxidizes sulfur, and the other is a pathogen, all related to each other? How did that evolutionary change take place? They must not be very big genetic changes. So how does that work? It works because all the phenotypes in this group are based on the electron transport chain. And this is how I look at the electron transport chain. I'm not John Olson, who can visualize all these three-dimensional things in the cell membrane. I have a very simplistic view of it. The electron transport chain is an electrical device. It sits in the membrane, and it does a very cool thing, which is it separates oxidation-reduction reactions into independent half reactions. Think back to your Chemistry 101. Oxidation and reduction reactions can always be divided into an oxidation reaction and a reduction reaction. And it's just electrons that separate those two things. That's how this works. And so you have an oxidation reaction in one place and a reduction reaction in the other. And the electrons that are released by the oxidation are captured, fed through the electron transport chain, and run the reduction reaction at the other end. And so the electron transport chain represents a series of electron carriers where the electrons are passed from one thing to the next down this chain. But it's not quite so simple as that. There's actually electron carriers and hydrogen carriers. So a hydrogen is just an electron and a proton, right? And during this electron transport, they alternate between electron carriers and hydrogen carriers. And so it's quite common in there for an electron carrier to donate an electron to a hydrogen carrier, and which of course then has to pull a proton out of the environment to make that hydrogen. And then it transfers it to a hydrogen or electron carrier where the electron is donated and the proton is released. And this goes back and forth over and over again in electron transport. And it's physically arranged in such a way that when, for example, right here, a hydrogen carrier donates to an electron carrier, that happens on the outer surface of the transport chain. And that proton then is released into the outside of the cell. But when an electron carrier transfers to a hydrogen carrier, that happens on the inside. So when it needs to pull a proton out of the environment, it pulls that proton out of the cytoplasm. The result is that as the electrons transport through this thing, protons are taken from the cytoplasm and released into the outside. And so you have a proton pump being driven by these oxidation-reduction reactions. The result of this is high concentration of protons on the outside, low concentrations on the inside. That is, acid on the outside, base on the inside. It's not actually, by the way, most of the energy of this is not from the chemical gradient of protons or from the acid-base difference between them. Most of the energy of this proton gradient is electrical. Net positive outside, net negative inside, separated by a thin membrane. Think back to your physics. What is this called? It's a kind of potential. It's electrical potential. This is a capacitor. That's all it is. It's like a Leyden jar to store energy. That energy, of course, is harvested by ATPase to make ATP. All of the phenotypes that we talked about in this group use this same process. The difference is, what is this oxidation half reaction and what is this reduction half reaction? If this is the oxidation of NADPH, then the organism is a heterotroph. It's getting its electrons from organic material, And if this is oxygen, then it's a standard aerobe, E. coli. If this is the oxidation of sulfate to sulfide, then this is a sulfur reducer, like desulfovibrio. If this is a purple photosynthetic organism, then this is the photoreaction center here and here. Here where it donates the electrons after absorption of light, and here where it's recycled again. And so this is also the basis of photosynthesis. If this is an iron oxidizer, then oxidized iron goes here, and then whatever your reductant, like a reducing reaction like sulfate, is there. And so what these organisms have done is figured out how to modularize the two ends of this chain. So that any number, in order to change, for example, from a standard aerobic heterotroph to an anaerobic heterotroph, all you need is one protein. The protein that sits here and oxidizes something other than, reduces something other than oxygen. For example, nitrate, and now or sulfate, and now you're carrying out anaerobic respiration. Yeah?
Don't you need like acidic proteins to assemble your protein, like nitrogenase or phosphatase?
So nitrogenase is more complicated. In general, these are single proteins. And all they do is catalyze this half reaction. And they do so docked to the electron transport chain. Now, if there's specific weird carriers, if there's an iron sulfur center in there, then maybe you need another enzyme to build that iron sulfur center, but not usually. So the trick here is to make the ends of this transport chain generic docking stations, to dock whatever enzyme you need to run a half reaction for electron transport. That's it. And so horizontal transfer of a single gene can completely change the phenotype of the organism. That's all it takes. In some cases, it can be more than that. For example, the photosystems, if you've got to have all the photosystem stuff for that. But in most cases, you're talking about one or two proteins to go across a huge range of phenotypic diversity.